Hi everyone, my name is Megan Flynn and I will be covering the last section of Chapter 2 in our Concepts of Biology OpenStax textbook. So if you have had a chance to look at the other two sections, this will just be the continuation of the last section of the chapter. If you have not had a chance, I highly suggest you click the subscribe button so that you can find the videos, the previous videos, a lot easier. So the first two videos that I did on Chapter 2, our Chemistry of Life section in our OpenStax textbook, were on the building blocks of molecules and water. So now we're going to focus in on biological molecules. Okay, so this is, that next step, looking at how atoms make molecules, molecules make macromolecules, and so forth. So we're getting into, the building blocks of life. So by the end of this section 2.3, you will be able to describe the ways in which carbon is critical to life, explain the impact of slight changes in amino acids on organisms, as well as describe the four major types of biological molecules and understand the function of those four types of molecules. So we have quite a bit of information that is covered in this chapter, or this section of the chapter. So let's start out by just kind of refreshing our memory on carbon. So we are carbon-based, so living things are carbon-based. That means that we have a lot of carbon atoms in our body, and they're making up the major macromolecules of our body. And so let's just refresh our memory of what that carbon atom looks like. So remember, our carbon atom, if we look at the periodic table, its atomic number is 6. So that means that, if you remember from previous videos, it has 6 protons. So the atomic number always equals the number of protons. So it has 6 protons. And if it's just a regular neutral atom, then it also has 6 electrons. So if we're studying our carbon, we look at its electron shells. We know that the first shell is filled with 2 electrons, and then the second shell is filled with just 4 out of 8 electrons. So it only has 4 filling its valence shell, but it wants to have 4 more. And so because it needs so many more electrons to fill its shell, it's going to be forming covalent bonds with up to 4 other atoms. And remember, those covalent bonds, they are strong bonds because the 2 atoms are sharing the electrons. So here's an example of a carbon atom that is bonding with 4 different atoms, in this case hydrogen atoms. So we have 4 bonds, and each are sharing a pair of electrons, so that adds up to 8, filling the valence shell of the carbon. Okay. Now when we're talking about carbon bonding, we're going to be looking at the structure of macromolecules that are carbon-based. And because that, we'll notice when we're looking at carbon molecules or carbon compounds, they tend to form chains, like in this case, where we have a bunch of carbon lined up where they're bonded to each other and hydrogens. And then here we have a double bond with an oxygen and an OH. Or maybe they can be doesn't have to necessarily just be carbon and hydrogen or carbon and oxygen. It can also be other molecules. So here is an example of a carbon molecule, carbon-based molecule that has a nitrogen in it. Okay, and then our carbon-based molecules don't necessarily have to be linear. Okay, so it doesn't have to be branching linear. It can form a ring structure. So here you can see an example of carbons forming a ring connected to an oxygen here. And then attached to each central carbon, we have perhaps an OH hydroxyl group and a hydrogen. Okay, something like that. All right. So Now that we have a little bit of information about carbon, we're going to take a look at the biological macromolecules of life. So there's 4 of them, and they're all carbon-based. So the first one we're going to study is carbohydrates. So you may think of carbohydrates as like an energy source. You know, if you eat a lot of carbs, you get a lot of energy. Lipids, think of those as like the fats. They can store energy, but we also have other lipids in our body that we'll talk about. Proteins, they are the building blocks of a lot of our components of our cells. Think of like muscles and things like that. They do a lot, proteins, in our body. And then finally, nucleic acids. That's the 4th major macromolecule. And these are the carriers of genetic information. Okay, so it's used in heredity. So we're going to look through each one of these, and we're not only going to describe their structures, but we'll also describe their functions. Okay, so you'll get a chance to really learn the importance of each one of these macromolecules. So let's start with carbs. So the carbohydrates, well, if you take a look over here, there's a lot of carbs in this picture, right? And they are a good energy source. So we actually will break down complex carbohydrates into simple sugars, and those simple sugars will provide the chemical energy that our cells need to produce cellular energy, ATP. And we'll learn more about the importance of cellular energy, ATP, in subsequent chapters. But for now, the nutrient that brings us that source of energy is a carbohydrate, okay, most of the time. But it doesn't just provide energy. It's not just used for cellular energy or converted to cellular energy, but it also provides structural support. So we'll talk about, for example, how plants, they have really complex carbohydrates that make their cell walls. And we'll talk how that helps provide support and structure for the cells. So those are the 2 main functions of our carbohydrates. So providing energy as well as providing structural support. Now, one interesting thing about carbohydrates is they're always found, or the molecules are always found in this ratio where we have 1 carbon to 2 hydrogens to 1 oxygen. So for example, glucose is a simple sugar, and glucose, you may have heard about it. It's basically C6H12O6. So it has 6 carbons, 6 oxygens, and 12 hydrogens. And so it fits with that same ratio. So there's twice as many hydrogens in glucose than carbons and oxygens. So C6H12O6. So we have 12 hydrogens to 6 carbons and 6 oxygens. So our carbohydrates, they always follow this ratio. So that's one way that if we're looking at a chemical equation, we can distinguish a molecule as being a carbohydrate because it has this ratio to it. And as far as our carbohydrates, I keep talking about like simple sugars. So simple sugars are the smallest form of, or the smallest, it's a subunit that forms like a complex carbohydrate. And we call those simple sugars monosaccharides. So think monosaccharide, it's known as a monomer from the polymer, which in this case would be a polysaccharide. So monosaccharides, those are simple sugars. If we take 2 monosaccharides and link them together, we get a disaccharide. So think di means 2, mono means 1, saccharide, think sugar. So monosaccharide, a single subunit sugar. Disaccharide, 2 of those subunits connected together to form a disaccharide. And then poly means many. So you can guess what polysaccharides are. They're made up of a bunch of those monosaccharides linked together. So we'll go through some examples of each of these. So let's start with the monosaccharides. So as I stated before, mono, think 1. So this is just 1 subunit. And they're also known as the simple sugars. And here are some examples of them, what they look like. So we can tell right away that it is a carbohydrate because it follows that ratio. There's twice as many hydrogens to oxygens and carbons. So glucose, this one's an example of glucose here. It has 6 carbons. It has 1, 2, 3, 4, 5, 6 oxygens. And if I counted them all up, I would see there's 12 hydrogens. Okay, so it follows that ratio of twice as many hydrogens to carbons and oxygens. So that's one characteristic. When we are talking about a monosaccharide, they can have as little as 3 carbons in there and as many as 6. So that's typical, okay? So often we'll see them as 6 or 5, but they can have, less than that. They can have more, but it's more rare. And these are basically what will provide a chemical energy source for our cells. So our cells will actually take in this molecule, this chemical energy form, and convert it to cellular energy. So this is why it's an energy source for our cells. Now, here are 3 examples of monosaccharides. So we have glucose, galactose, and fructose. And one interesting thing about these 3 molecules, they all share the same chemical formula. They all have 6 carbons, they all have 12 hydrogens, and they all have 6 oxygens. But notice they have slight differences, okay? So you can see between glucose and galactose, you can see the placement of the hydroxyl groups, the OHs, is slightly different. Okay, so you have like 4 on this side and 1 on that side. Here you have 2 on this side and 3 on that side. Fructose is a little different. Where its double bond is. So glucose and galactose, the double bond is on the first carbon. Here it's on the second carbon. Okay, so that changes kind of what it looks like. So these are considered isomers, okay? Now, if we take 2 of these monosaccharides and put them together, okay, with a chemical reaction, they will form a disaccharide. So remember, di means 2, so 2 sugars together. And the way that happens is through a chemical reaction known as dehydration synthesis. So dehydration, that sounds like, well, you're dehydrated. If you're dehydrated, you don't have enough water, right? So think of dehydration synthesis is removing water to form a bond, okay? So let me show you what I mean here. Here we have, let's say this is glucose and this is galactose. They have the same formula, so I can't really tell directly from this. But, I'm just going to use this as an example here. So let's say that we have, or actually it says here sucrose. So sucrose is a glucose and a fructose. So I guess I'll use these 2 instead. So we're going to take this molecule and this molecule, and we're going to bind them together. And they're going to bond together with a covalent bond. And in doing so, once they're bonded together, an OH from one of the molecules and an H from another of the other molecule will bind together to form water. So let's take a look over here. So let's say that we, in this case, we have the OH from glucose is removed and binds with the H of this fructose, okay? And in doing so, now these 2 molecules, these carbons will link together in a covalent bond. In this case, it's a type of covalent bond known as a glycosidic linkage, but that gets into the fancy words you probably don't need to know. But it's just a covalent bond that binds these 2 molecules together. In order to do that, a water molecule is going to form by removing an OH from one of the molecules and an H from the other. So if I remove this OH and this H and bring them together, they form water, okay? And then glucose and fructose are brought together with a bond with that water removed. And so you can see that combined, their chemical formula is a little different now, okay? So we have our 12 carbons, so that's basically adding those two numbers together. But look at the hydrogens, okay? So if we added these two together, we would get 24 hydrogens. But since I removed 2 of them, it dropped down to 22 in our sucrose chemical formula. And then same with oxygen. We have 6 oxygens plus 6 oxygens is 12, but I've removed 1 of them to form 11, okay? So here's the chemical formula of our disaccharide table sugar. We've removed that water and we've formed that disaccharide. And this is through a dehydration synthesis reaction. So we're removing water in order to bring 2 monosaccharides together. Now we can continue having all of these dehydration synthesis reactions occur to bring monosaccharides together to form a long chain of monosaccharides. So this is what we call a polysaccharide. So think poly as many, many sugars. So it's just a bunch of monosaccharides stuck together. And you'll notice here we have like a ring structure. So glucose, it actually forms rings. I know in the last slide we saw them as in a linear structure, but they actually do form a ring. And in doing so, they look kind of like this, okay? And so we have one glucose attached to another glucose with a covalent bond, and another glucose attached with another covalent bond, and so forth. And so we get these linked monosaccharides to form the polysaccharide. So the main examples that we give of polysaccharides are starch, glycogen, cellulose, and chitin. So it is because these are the most often present in our cells, whether they're plant cells, animal cells, fungus, so forth. So let's take a look at starch. So starch is found in plants, and it's an energy storage for plants. So if you take a look at this starch molecule, it's just a bunch of glucose molecules linked together. So if that plant stores away starch, it has a stored energy source within it. Okay, that helps when this plant needs to have can't do photosynthesis 24 hours a day because it's dark out, and so it can rely on these stored resources. Okay, so these starch molecules can end up being formed inside the vacuole of the cell, and we'll talk about the cell structure in a bit, or in the next chapter. But you can see it's just glucose linked together. That's all it is. Glycogen is kind of starch for animals. Okay, so it's a way that we store glucose in our bodies. So glycogen is just a complex carbohydrate or a polysaccharide of glucose. So there's a bunch of glucose molecules. It looks a little different from starch in that it kind of does a branching, but it's basically a storage of glucose. So when we need glucose, we can break these bonds and get that glucose molecule in order to undergo cellular respiration and get our ATP cellular energy. And we'll talk more about that later on in subsequent chapters. But so this glycogen molecule tends to be common in our muscle tissues. So that muscle tissue has quite a bit of glycogen, which makes sense because your muscles need a lot of energy. Okay, you're constantly constricting them or moving them and things like that, so they need a constant source of chemical energy in order to convert that to cellular energy to move and contract and things like that. And so here is that source. Okay, so glycogen is like our stored sugars within our cells, and it's common in certain areas of our body. Cellulose. Cellulose is actually one of the, if not the most abundant organic molecule on Earth. So it is what makes up the cell wall of our plants. And there's a lot of plants out there. And you can see that it's actually the same thing as I said before. It's a bunch of glucose, but it looks a little different than starch. Okay, so you can see on starch, we have, you know, the OH group all on one side. Here it's kind of opposing sides. Okay, that's it. But cellulose makes up the cell walls of our plant cells. And cell walls, we don't have cell walls as animals, but plants, they have a cell wall that surrounds the plant cell. And that cell wall is going to allow the plant to have structure and protection. Okay, so that cellulose is what provides that structure and protection for that plant cell. So that is another example of a polysaccharide. In this case, it doesn't provide any energy, but it provides structural support. Okay. And then the last one is chitin. So chitin is another type of polysaccharide. It's a little different from the other three in the sense that we have nitrogen involved there. Okay, so it has a different element within it. And this is a unique type of polysaccharide that helps make up the exoskeleton of many of our arthropods. So think of, you know, insects like ants or something like that. Their exoskeleton is composed of this chitin, so it provides structure and rigidity. Okay, so those are examples of polysaccharides. So we've talked about the carbohydrates as one of the 4 macromolecules. So now we're going to move into the second one, the lipids. Now lipids, they are pretty interesting category of macromolecules. They don't actually have any monomers or subunits that are consistent amongst each other. And so their structures are very unique amongst the different lipids. So what makes them lipids is basically that they're hydrophobic. Okay, so if you recall from earlier, was it 2.2? And yeah, I guess mainly in 2.2, that video I discussed hydrophilic versus hydrophobic. So hydrophobic, hydro means water, phobic means fearing. So these are water-fearing molecules. And they're water-fearing because they're made of nonpolar molecules. Okay, so if it's nonpolar, it's not going to want to bond with water. And so since it's not wanting to bond with water, it's not going to dissolve in water or anything like that. And so that's what makes lipids so unique. They're basically just hydrophobic nonpolar molecules. Okay, so it's a little different than carbohydrates, where we have monosaccharides form disaccharides and polysaccharides. This is just like a unique category of a bunch of molecules that have the same property, which is they're hydrophobic. Okay. And they do have quite a number of functions within our body. They are an energy source. So lipids being nonpolar, they're often, carbons and hydrogens linked together. So we can use that as a stored energy source. We can also use it as insulation. So if you think about your own bodies, we need to insulate or cushion our organs. So if we're going to go, do jumping jacks or something like that, we don't want our liver banging into our heart. That would damage, our vital organs. And so we actually have lipids covering them. So they provide this, provide insulation and protection for our organs. Some of our hormones are actually lipid-based. So cholesterol, for example, we'll talk more about that, but cholesterol is a hormone, is actually a precursor to our sex hormone, testosterone, estradiol. And hormones, they're chemical messengers within our body. And then the final function that we can talk about is that they make up the bulk of the plasma membrane of our cells. So if you think of our animal cell has, this barrier, and that barrier is made of a lipid-based molecule, okay, phospholipids. Okay, so some examples of lipids. I always think of lipids as fats, okay, because that makes sense. So in our diet, you can think of like fats as lipids. Oils are a type of fat. Waxes, so think of like beeswax, things like that. Phospholipids, they're what makes up the plasma membrane. And then steroids, they can be the precursors or even be the hormones themselves within our body. So we're going to talk about more about our lipids and their function. But I just want to show you, look at this cute little river otter. So the river otter, you can, he's very cute, but he spends a lot of time in cold water. And to protect him from that, he actually has, very thick fur, but he also has kind of a bunch of lipids that kind of coat the fur to prevent too much hydration into the structure of the fur. So it actually creates kind of this waterproofing for our friend here. Okay, and he's so cute. All right, so let's take a closer look at the structure of different types of lipids. So as I stated before, the first thing I think of when I think of a lipid is a fat. So a fat molecule consists of glycerol and fatty acids. So if we take a look over here, we can see here is a structure of a saturated fatty acid. So basically we see the carbons linked together, and then each carbon is linked to another carbon or two other carbons, but it's also linked to two hydrogens. Okay? And so it's just basically a bunch of C's and H's. Okay? And then here we have, this is a carboxyl group where we have a double bond to an O and a bond to OH. So if we take fatty acid and we link it to a glycerol, so glycerol is basically just this part, which is again just a bunch of C's and an H. Okay, we can form a triglyceride. So a triglyceride, tri means three, so it's three fatty acids linked to a glycerol. So the fatty acids being, this kind of structure with the carboxyl group and the carbons and hydrogens in a chain. You can see how that carboxyl group combines with glycerol, is just a bunch of C's and H's, and to form a triglyceride. Okay? So three fatty acids in a glycerol. And then if we take a look at these two fatty acids, saturated versus unsaturated, what do you notice is different between them? Okay, they both have kind of the starting structures the same. Okay, they have a carboxyl group with a bunch of carbons and hydrogens, but then look at what's going on over here. In the unsaturated fatty acid, we have a double bond, two lines, double bond between two carbons. And because of this, we actually have less hydrogens. Okay, so if we look up here, for example, we have a carbon and a carbon. This carbon is bonded to another carbon, so there's two of its valence electrons, and then the other two are connected to hydrogen. If we look over here, we have a double bond between the two carbons, and with that double bond, okay, we have four electrons shared between the two carbons. Here's another two shared between the other carbon. So that just leaves room for one hydrogen to attach. So instead of two hydrogens, we have only one. So unsaturated fatty acids, they contain a double bond, and they don't have as many hydrogens. So think saturated fatty acids as saturated with hydrogens, lots of hydrogens attached to each of the carbons. Unsaturated, we have less hydrogens, and the reason for that is because we have a double bond. Okay? So these are examples of fats within our body, or our foods, or whatever we're talking about. It's a type of lipid. Now in nutrition, we think of like unsaturated fats. They're common in like our vegetable oils. You know, they're basically unsaturated fats tend to be liquid at room temperature. So like olive oil, canola oil, those are all examples of unsaturated fats. Saturated fats, they tend to be solid at room temperature. So think of butter, okay? Butter, if you have a piece of meat, there's like solid fat in there, things like that. So it would be solid at room temperature. Now, our food industry created a way that we can have something called a trans fat. So what a trans fat is basically taking an oil, which oils, they're plant-based, okay? They're unsaturated fats. They're liquid at room temperature. They're plant-based. But they're making it a solid, okay? So if they're making it a solid form, but it's an oil, then they have to change the structure of the molecule. And so they can do that through a process known as hydrogenation. So in hydrogenation, they're going to kind of change the structure to basically create a change in the way that the double bond is oriented. So you can see here's a normal fat, unsaturated fat molecule. So we have a double bond between the two carbons. And notice where the hydrogens are. They're on the same side. Okay, so they're both together on the same side. So cis thinks same. Cis thinks same side. Trans think opposite sides. So the only thing here is that we still have that double bond, but look at the placement of the H's. They're on opposite sides. So a trans fat molecule is chemically different. Okay, so instead of having a bent shape that makes them more liquid, they tend to be more linear shape. So they tend to be solid at room temperature. So think something like margarine or shortening. So margarine and shortening, they're made from vegetable oils, okay? And basically they've taken those plant oils and then they've converted them into trans fat to form a solid form. Okay, some peanut butters are like that. So if you get the natural peanut butter, you'll notice that it separates, right? You have like the solid part and then you have the oil part on the top. So that doesn't contain any trans fats because it's, you know, oil-based, unsaturated fats. If you have a peanut butter that is solid all the time, you know, think Skippy or something like that, then it contains those trans fats, okay, to make it solid instead of separating into the oil part. So trans fats, they've been very popular in the past, but recently we found that they actually do cause some issues with our body, particularly with our cholesterol levels. So if you've heard of cholesterol, you know that when they measure cholesterol in your blood, they're looking at the LDLs versus the HDLs versus the triglycerides. Okay, so we already know what a triglyceride is. So that's the fatty acids linked together with a glycerol. The difference between the other ones, the HDLs and the LDLs, so think LDL is it's a low density lipid versus a high density HDL lipid. And when we're looking at our cholesterol level, we want to have higher numbers of HDLs to lower numbers of LDLs. Okay, because that means that it, like HDL is kind of what we consider the good cholesterol, LDL is kind of the bad cholesterol. And so, we want more of the HDLs, less of the LDLs in our overall numbers. And so trans fats, they tend to increase our LDL number. Okay, so if we're increasing our LDL number, it could, be a precursor to heart disease and things like that. So trans fats, they're causing we're finding out that they're causing some health problems, and so that's why you see a lot of big movement in nutrition to get away from the trans fats. So moving on, we've talked about lipids in the form of fats and oils. So now let's get into the phospholipids. So phospholipids, as I stated before, they help make up our cell membrane. So our cell membrane that surrounds the cell, it's going to be what we call a semi-permeable membrane. So it has to provide protection for our cell, but allow certain things in and out of our cell. So it's that barrier. And the reason why we have phospholipids is because phospholipids, they consist of a hydrophilic end and a hydrophobic end. So if you recall, hydrophilic means water loving or liking, hydrophobic means water fearing. And so here is our example of a phospholipid. Now, think phosphorus. So there's a phosphate group over here. That phosphate group over here is going to be polar. And you can even say, hey, look, there's a little negative there. So it's going to be polar. And because it's polar, it's going to be hydrophilic. That phospholipid is connected to two fatty acids. So fatty acids, they're nonpolar. And since they're nonpolar, they're going to be hydrophobic. So when we're talking about a phospholipid, they have that unique property of having a hydrophilic head and two hydrophobic tails. Okay, and so that's going to be important when we're allowing certain things to enter the cell and certain things to not enter the cell. And we'll talk more about the cell membrane in subsequent chapters. All right, so the last kind of group of lipids that we're going to talk about are steroids. So steroids, what's unique about them is that they're ring shaped. Okay, so they have a ring structure. And typically it's about 4 carbon rings. And so you can see a big difference between a steroid versus all the other lipids here that have these kind of linear chains to them. And this is an example of cholesterol. So cholesterol is, we talked about earlier, as being important component of our blood that could potentially lead to heart disease if we have too much of one, too little of the other. But cholesterol actually has a lot of importance to them as being a precursor hormone. So our sex hormones like testosterone and estradiol are cholesterol based. Okay, so they look very similar to this structure, but they have some additional functional groups to them. Also, cholesterol is a small component within our cell membrane. So our cell membrane is mainly made of phospholipids, but we do have cholesterol in there, and that helps stabilize it. And we'll talk more about that when we get into the cell membrane. So this, again, being another type of lipid, it's hydrophobic. It can't be, dissolved in water, and it has, the same or very similar structure or chemical formula. So a bunch of C's and H's and O's, but in this case, it's in a ring structure. So we're going to watch a video on this.
Diseases and are bad. In fact, most fats in adequate amounts are required for normal bodily functions, especially brain functions. There are also good fats that actually decrease the risks for cardiovascular diseases. A fat molecule is composed of a glycerol head and 3 fatty acid tails, each of which is a long hydrocarbon chain, a carbon skeleton bound to hydrogen atoms. When all the carbons are fully bound to hydrogens, the fatty acid is said to be saturated. All the bonds between carbon atoms are single, and the hydrocarbon chain has a straight shape. A fat molecule made entirely of saturated fatty acids is a saturated fat. Due to their straight tails, saturated fats are compact and solid at room temperature. On the other hand, when the hydrocarbon chain has fewer hydrogens, it is said to be unsaturated. Instead of binding to a maximum number of hydrogens, some carbon atoms bind to each other via a double bond. The presence of double bonds may bend the hydrocarbon chain, creating gaps between molecules, making them less compact. As a result, unsaturated fats are usually liquid at room temperature. A fat molecule that contains only one double bond is a monounsaturated fat, while one that has multiple double bonds is polyunsaturated. Dietary fats provide fatty acids for the synthesis of the cell membrane, a vital component of all animal cells. The gaps in unsaturated fatty acids provide membrane fluidity, facilitating membrane transport and cellular signaling. While both types of fats are needed for an optimal composition of the cell membrane, too much saturated fat, which is commonly the case in a typical American diet, would make the membrane rigid and hinder cellular responsiveness. Membrane fluidity is most important in the nervous system, where neuronal response requires extremely fast cellular communication. A certain ratio of unsaturated to saturated fatty acids is also required for the formation of myelin, the insulating material that wraps around axons of neurons and speeds up the conduction of electrical signals. The body is capable of synthesizing all the fatty acids it needs, with the exception of polyunsaturated fatty acids omega-3 and omega-6, which must be obtained from the diet. These are known as essential fatty acids. In general, unsaturated fats are healthier than saturated fats. Unsaturated fats decrease the risks for heart disease by reducing the amount of bad cholesterol, LDL, and increasing the good cholesterol, HDL, while saturated fats increase both good and bad cholesterol. But not all unsaturated fats are equal. In fact, a type of unsaturated fat, known as trans fat, is the unhealthiest of all. A double bond can give rise to 2 possible configurations, cis and trans. Cis is when the 2 hydrogen atoms are on the same side of the bond, while trans is when they are on the opposite sides. A cis double bond bends the fatty acid molecule, while the somewhat more symmetric trans configuration does not. A trans fat is therefore similar in structure to a saturated fat. More importantly, trans fats rarely occur in nature, so the body does not have the necessary enzymes to break them down. Diets rich in trans fats increase the bad cholesterol LDL and reduce the good cholesterol HDL, having the most detrimental effect on blood vessels. Trans fats are found mainly in partially hydrogenated oil products, such as margarine. Because unsaturated fats are less stable and spoil faster, food manufacturers add hydrogens to make them more saturated through a process known as partial hydrogenation. This process not only prolongs shelf life of vegetable oils, but also turns them into solid or semi-solid products, which are preferred by commercial bakers for their low cost and wide range of different textures. Unfortunately, partial hydrogenation also converts some of the cis double bonds into trans configuration, producing trans fats. The FDA has officially banned production of partially hydrogenated oils in June 2018, but products made earlier may still be in use until January 2020. Thank you for watching. If you like our videos, be sure to check out our new page.
All right, so this video, by the way, is you can access directly from the OpenStax Concepts of Biology textbook. There's a link to it in section 2.3.
Yeah, Pete.
So we learned about the different types of fats, as well as, you know, good and bad unsaturated versus saturated versus trans. Just realize that even though they stated that saturated fat is less healthy than unsaturated fat, we still need fats in our body to produce those plasma membranes. So as long as we have it in moderation, everything seems to be okay. We just want to try to avoid the trans fats. As far as our next macromolecule, proteins, they have kind of going back to this similar structure as a carbohydrate. So remember carbohydrates, we have a monosaccharide, and then you get two monosaccharides linked together to form a disaccharide, and then multiple monosaccharides linked together to form a polysaccharide. So proteins, they go along the same kind of pattern. And they're actually the monomer of a protein is known as an amino acid. So a protein is made of a bunch of amino acids linked together. Okay? So proteins are one of the most important types of macromolecules because they have a lot of different functions within our body. So I always think of proteins as like the building blocks of our body. We have our proteins do a lot inside of our body. So we have some that are part of structural support, such as the cytoskeleton within of our cells, keeping the shape. Regulatory, so allowing certain items to move into or out of the cell itself through a transport protein. Contractile, our muscles are made of myosin and actin, and they can expand and contract in order for movement to take place. Protective, so within our cells we have these structures, which we'll get into later, called lysosomes, and they provide protection from like bacteria and things like that. So they're part of like immune response. Transport, so we have special transport proteins that can move structures or move other molecules from one area to another. They're involved with storage, so storage of energy and things like that. So think of like a chicken egg. Inside that chicken egg, the yolk part is where the embryo is, if it's fertilized, and then the egg white is made up of a bunch of albumin. Albumin is protein, so it's storing a bunch of amino acids for the developing chick. Enzymes, a lot of enzymes, actually most of our enzymes are protein based. So enzymes, they are catalysts of our reactions. So they help speed up reactions. And we'll talk a lot about enzymes in the subsequent chapters. And then hormones. So we talked about cholesterol, a lipid, being a precursor to some of our hormones. Most of our other hormones in our bodies are protein based. So for example, think of insulin. So insulin is going to help regulate sugar in our blood, and allowing cells to absorb or get rid of or not accept sugar. So the hormones are just kind of chemical signals within the body. So these are just some of the different functions of our proteins, so very diverse. Now, as far as our proteins, the monomers that make up the proteins are called amino acids. So amino acids, they have kind of the same backbone to them, but then there's like differences in individual amino acids. So all amino acids, they have a central carbon bonded to an amino group and a carboxyl group. So if you take a look, here's 5 different amino acids, and all of them have a carbon in the center, and that carbon is attached to an amino group. So amino is nitrogen with 2 hydrogens, and on the other side a carboxyl group, which is a carbon double bonded to an oxygen, and then the OH group is also bonded to the carbon. Okay, so the backbone is the same for all amino acids. They all have a central carbon, they all have an amino group, and they all have a carboxyl group. Okay. The only real difference is what we call the R group, or sometimes you call them the side chain. So the R group, it could be, is different amongst the different types of amino acids that we find in nature. So you can see here we have an amino group, or excuse me, an R group, where we have a C and 3 H's. Okay, that's a methyl. Here we have another amino group where we have a C that's bonded to two other carbons and so forth. Here we have a carbon and it's bonded to a nitrogen and so forth. So the R group is the only difference between amino acids. The backbone will be the same. Okay? And when we're talking about the R group is going to help determine how that particular protein functions. And we'll talk more about that when we talk about how a protein folds its structure. Because some of these R groups, they are hydrophilic, some of them are hydrophobic, some of them are more acidic, some of them are more basic. And so, some of those just key differences can affect how the protein functions. Okay. Now, if we take two amino acids and link them together through a dehydration synthesis reaction, we're going to form it's called a dipeptide, which, if we have a bunch of amino acids together, it forms a polymer called a polypeptide. So peptides are another word for amino acids. Okay, so the subunit being a peptide or an amino acid. And when we have two peptides held together, we get a dipeptide. If we have 3 or more peptides together in a line, we get a polypeptide. So think poly, many, peptide amino acids. And so they're basically all linked together with a covalent bond. Just like with carbohydrates, that covalent bond has a special name to them. They're called peptide bonds. And so the way that happens is we have in our backbones here, so let's say we're combining alanine with valine. So what's going to happen is we're going to have a dehydration synthesis reaction. So remember, in a dehydration synthesis reaction, we're going to take away an OH from one of our molecules and a hydrogen from another molecule to form water. And by doing so, then we link the two molecules together with a covalent bond. So imagine me taking this OH away and one of these H's away. So the H and the OH combine together to form water. Now we have the rest of the molecules will join together in a covalent bond. So now we have two amino acids linked together with a covalent bond. And it's always the backbone, okay? It's this part that's bonding. It's not the R chains, okay? So that'll be, again, important when we're talking about protein folding. So speaking of protein folding, our protein structure, we actually have 4 levels of our protein structure. So when we're just talking about the links, okay, so here we have each one of these little beads are individual amino acids, okay, and they're linked together with a peptide bond, just like what I talked about earlier. If I remove the OH of the alanine and combine it with the H of the valine, we get water, and then now we have alanine and valine connected together to form a bond, a covalent bond. Okay, so that's a bunch of these amino acids linked together. So that's the primary protein structure. It's just a sequence of amino acids in a chain. And in our protein, we can have 100, we can have thousands of these amino acids linked together. So it's a bunch of alanines and valines and all of these linked together in a chain. So that's the primary structure. Now, we'll start to see the chain fold. And the way that it's folding is going to involve hydrogen bonding. So let me go back to the previous slide. It'll let me go back. Hydrogen bonding. So if you notice in our structure here, we have, this H sticking out. And so this H can start maybe being attracted to an OH, or, an electronegative of another nearby molecule. So because we're involving two nearby amino acids that are not directly connected by covalent bonds, we end up seeing the chain kind of fold, okay? And it can fold one of two ways. So it can fold as like an accordion. So we end up with this beta pleated sheet, or it can kind of fold in a way that we end up with a helix. Okay, so we call that the alpha helix. So these are due to hydrogen bonding of the peptide backbone. Okay, so we see kind of the first fold. The tertiary structure involves those R groups. Okay, so we've already folded. Now we're involving the R groups. Now, as I stated before, the R groups, they have different properties to them, right? So some of them could be non-polar, some of them could be polar, some of them could be acidic, basic, things like that. And because of that, they're going to affect how the protein starts to fold even more. So the R groups that are hydrophobic, water-fearing, they're going to want to be closer towards the middle. So they're going to kind of fold inward. The hydrophilic, they like water, so they'll be kind of closer to the outside of the protein, and so forth. Okay, so those are just examples of ways that the beta pleated sheets, or the alpha helix, they start to kind of fold in a way that allows for the properties of the R chains to come in to balance here. Okay. Now, once we've reached the tertiary structure, this could be the end. This could be a protein ready to do whatever the protein is designed to do. Some proteins are not made of just one polypeptide. They're made of multiple polypeptides. And so that's when we get to this quaternary structure. So quaternary structure consists of more than one amino acid chain. So we might have one kind of tertiary structured protein over here. Here we have another tertiary structured protein over here, and then they're interacting together. So again, the name protein could designate the tertiary structure, or it could be the quaternary structure, depending on how many polypeptide chains there are. Okay, so now we have a fully functioning protein, whether at this level or at this level. Because of the complexity of the folding, the protein needs to be in a certain environmental condition. If we deviate from that environmental condition, we can end up causing that protein to unfold. And so that's called denaturization, okay, or denaturing the protein. So it loses its shape in a protein. And some of these environmental factors could be temperature changes, change in pH. We talked about pH in the last video. Exposure to certain chemicals. These can cause the R chains to kind of break away from the others and unfold. So if we have an unraveling of a protein, then the protein can no longer function like it's supposed to. So that's that process of denaturation. All right, so now we're on to the last of our macromolecules, the nucleic acids. So the nucleic acids, they're biological macromolecules that carry genetic information. They also could carry the instructions to build things within a cell. So when you hear nucleic acids, you might think right away DNA. So DNA is a type of nucleic acid, and it stands for deoxyribonucleic acid. So deoxyribonucleic acid, DNA. The other type you may have heard of is RNA, so ribonucleic acid. Okay, so we'll talk in detail about these two molecules when we get into our units on heredity. But for now, these are the macromolecules, and each of these macromolecules are made up of subunits, okay, the monomers. In this case, the monomers are called nucleotides. And nucleotides, they're made of three components to them. So they all contain, so DNA and RNA, they contain a phosphate group, a sugar group, and a nitrogenous base. So if we're talking about DNA has a, which this one is, we have a phosphate group, a 5-ring sugar, and then a nitrogenous base. And you can see it's nitrogenous because there's a bunch of nitrogens there. RNA, very similar. They have a phosphate group, a different type of sugar. Okay, it's still a 5-ring sugar, but it's got an extra OH down here. So it's ribose sugar, and a nitrogenous base attached. So the only real difference between the two is deoxyribose sugar in DNA and then ribose sugar in RNA. And then we also have some differences in the nitrogenous bases. Okay, there's actually 4 different types. For DNA, there's 4 different types for RNA. Okay, so these are the subunits that make up the nucleotide. Okay, now if I have 2 nucleotides brought together, they will link together, and the way that they link together is, let's see if I have a better picture. I don't think I have that great of a picture here. I don't. They're linked together by the phosphate group of one nucleotide and the sugar group of another nucleotide. Okay, so basically there'll be a bond between the phosphate group and the sugar group of 2 nucleotides. Okay, so basically you end up having this part. If this was another sugar and a phosphate group and a sugar and a phosphate group, this is the backbone of our nucleic acid. Okay, and so in something like DNA is a double helix. You see here this line, these indicate the backbone. Okay, this is the phosphate of one group bonded, covalent bond to a sugar of another nucleotide over and over again. Okay, so they're held together by covalent bonds. So the backbone here is very, very strong. So we have that holding together the strand. Okay, so here we have DNA. DNA is double-stranded, so there's 2 strands here. So this strand right here is held together by bonds between the sugar group of one nucleotide and the phosphate group of another. Okay. In between the strands, we have another bond. Okay. And this involves the nitrogenous bases of opposing strands. So here is a nitrogenous base sticking out from this strand. Here is a nitrogenous base sticking out from this strand. Okay, so these 2 nitrogenous bases can form a weak hydrogen bond between themselves. Okay, so this is what kind of makes up the double helix structure. So we have the backbone, which you can think of like the legs of the ladder, are strong. Okay, so they're not going anywhere. We have the sugar phosphate backbone held together by covalent bonds. The rungs of the ladder are held together by hydrogen bonds, and these are weaker bonds. Okay, so they're just, and these are holding the 2 strands together. Okay, and we'll get into the importance of this when we again get later into the chapters, when we go into genetics and DNA replication and protein synthesis and all that fun stuff. Okay, so as far as chapter 2.3, we're going to go through some review questions and then we're done with chapter 2. So let's start out with an example of a monosaccharide. Is it fructose? Is it glucose? Is it galactose? Is it all of those? So what would be an example of a monosaccharide? Remember, mono means one, saccharide means sugar. So we're talking about simple sugars. So which one of these are simple sugars? Is fructose simple sugar? Yeah, it's that C6H12O6 molecule. What about glucose? Yep, that's also C6H12O6, but differently structured. And how about galactose? Yep, that's still C6H12O6. These are all monosaccharides. So all of the above. Okay, and then just remember, when we combine two of them together, we get a disaccharide. Here's another review question. Cellulose and starch are examples of. Now, are cellulose and starch, are they monosaccharides? Are they disaccharides? Are they lipids? Or are they polysaccharides? If you chose polysaccharides, I would agree with you. So remember, starch is just a bunch of glucose units linked together and provides a storage of sugar. So in this case, like potatoes have a bunch of glucose inside them, stored away as starch. Cellulose, they make up the plant cell walls. And that is, again, it's just glucose monomers stuck together. So these are all examples of polysaccharides, because we're taking monosaccharides, glucose, we're linking them together in a long chain. So poly. Here's another review question. So this one, it says phospholipids are important components of, well, are phospholipids important components of the plasma membrane in animal cells, the ring structure of steroids, the waxy covering of leaves, or the double bond in hydrocarbon chains? So what are these important components of? If you chose the plasma membrane of animal cells, I would agree with you. So here's an example of a phospholipid. Remember, the phospholipid has a hydrophilic head and hydrophobic tails, okay? And so they're going to, when we have a phospholipid bilayer, we have two of these phospholipids where the fatty acids are facing each other and the hydrophilic heads, phosphate groups, are facing outward. So they make the plasma membrane. And then we have the monomers that make up proteins are called, are they nucleotides, are they disaccharides, are they amino acids, or are they chaperones? If you chose amino acids, I would agree with you. So remember, amino acids, they are linked together to form the primary structure of a protein, and then you have the primary structure folding into the secondary structure, and then that folding into the tertiary structure and so forth. So here you see like the little helix, secondary structure, and so forth. All right, so we are done with chapter 2. At the end of this section, you can go back now and be able to describe the ways in which carbon is critical for life, explain the impact of slight changes in our amino acids in organisms, think of the denaturation, describe the 4 major types of biological macromolecules, so you can describe what a carb is versus a protein versus a lipid and versus a nucleic acid, as well as describe their functions. Okay, what do nucleic acids do compared to carbohydrates in our body? Okay, so this completes chapter 2. So in my previous videos, I covered the other 2 sections of chapter 2. So if you didn't get a chance to look at those, you can look at those under our playlist. Easiest way to find them is just click subscribe, and then you can locate them on the YouTube channel. So now you should have a good understanding of the chemistry of life. In the OpenStax textbook, under chapter 2, there are additional components to the chapter, such as a chapter summary that kind of gives you a summary of all 3 of these sections, as well as key terms lists. So you can make flashcards if the terminology is a little confusing for you to remember. I've gone over the review questions already for each of the units here, but there's critical thinking questions in the chapters. You might want to go back and take a look at those critical thinking questions and see if you can answer them based on all of the information and hopefully understanding of the concepts throughout these videos that you've been given. Okay, so the next video I'm going to do is I'm going to get into chapter 3. So chapter 3, we get into cell structure and function. So now that we know, what atoms are and molecules and macromolecules, we're going to start getting into organelles of the cell that make up that cell structure. Okay, so we'll get into that in the next chapter.